Hello and welcome to lecture 13 of our course on stochastic modeling of biological processes. In section 4.1, we simulated the behavior of 1000 molecules by computing the individual trajectories of every molecule using the stochastic simulation algorithm A8C8, which is based on the stochastic differential equation 411. To visualize our results, we divided the computational domain, interval 0L, into 40 compartments and we plotted the numbers of molecules in each compartment in figure 4.2b and in this video. In particular, most of the computed information, like the details of 1000 trajectories, was not used for the final result, which was presented as the spatial histogram. We visualized only 40 numbers, the number of molecules in each compartment, instead of 1000 computed positions of molecules. In this lecture, we present a different stochastic simulation algorithm for the simulation of molecular diffusion. We redo this example, but instead of simulating the 1000 positions of the individual molecules, we are going to simulate directly the time evolution of 40 compartments. To formulate the compartment-based approach to diffusion, we divide our computational domain interval 0L into k subintervals of length h, which is given as L over k. We describe the state of the system by the number of molecules of chemical species A in each compartment. To do that, we denote by A with subscript AI the number of molecules in the ith compartment where the index I is going from 1 to k. Diffusion is then modeled as the following chain of chemical reactions. I have put chemical reactions in quotes because there are of course no real chemical reactions involved in this model. Each of our variables, AI, contains the number of molecules of the same chemical species. However, it is very useful to write this model as a chemical system, because we can then apply the whole stochastic chemical reaction theory, which we learned in chapter 1. In particular, from the mathematical point of view, there is nothing new in this section on the compartment-based modeling. The presented model can be considered as another example of a stochastic chemical reaction model, so you could consider section 4.2 as another exercise to practice the mathematical techniques which we have discussed in chapter 1. The only new idea in this section is that we have to decide what is our reaction rate, which I denoted as the lowercase d in this chain of chemical reactions. My claim is that I can choose it according to this formula 414 in the lecture notes. The lowercase d is equal to diffusion constant d divided by the compartment size h to the power 2. So let me first illustrate that this is indeed the correct choice of this reaction rate constant by implementing our example from section 4.1. Since we have written our diffusion model as a chemical system, we can use the Gillespie algorithm to simulate the time evolution of the system. To do that, we need to specify the propensity functions. Here I would like to note that the two reactions which corresponds to the jump to the left and jump to the right from the ith compartment have the same propensity function equal to d over h squared times ai. Therefore, we do not need to compute it twice. In particular, we can simplify some steps of the Gillespie algorithm when we write it as a computer code. However, the resulting algorithm is just the Gillespie algorithm applied to this system of 2k-2 chemical reactions. Here we have k-1 reactions corresponding to jumps to the left and k-1 reactions corresponding to jumps to the right. This formulation implicitly implements the no-flux boundary conditions which we have in our model. There is no way to jump outside of the first compartment to the left or outside from the last compartment to the right. So the reflective boundary conditions have already been implemented in this diffusion model, and we do not need to worry about them. I have included the Gillespie algorithm applied to this chain of chemical reactions as the stochastic simulation algorithm A9F9 on page 103. The results of the simulation based on the Gillespie algorithm are presented in figure 4.3b on page 104 of the lecture notes, which I have included on this slide as the panel on the right. I compare it with the panel on the left, which was obtained by the diffusion model based on stochastic differential equations, which we have presented during our last lecture. However, if I did not tell you which model was implemented in which figure, you would not be able to guess this information. Considering diffusion only, both approaches provide the same information at the level of spatial histograms. But the underlying methods are different. On the left, I simulated 1000 individual molecules, while on the right, I simulated the time evolution of 40 compartments. In each model, I am comparing the results with the red line, which is the solution of the diffusion equation with no flux boundary conditions. The diffusion equation gives us the average behavior of the SDE-based model on the left. If I pass to the limit of the compartment size h goes to 0, then the diffusion equation also describes the average behavior of the compartment-based model of diffusion on the right. Here I should note that the limit of the compartment size h going to 0 is fine for the diffusion process, but it can be problematic when we start considering reaction diffusion processes, which we will discuss in detail in chapter 6. The compartment-based stochastic simulation algorithm A9F9 does not compute the trajectories of individual molecules. However, we can still compute a plot comparable with figure 4.2a. To do that, we repeat the simulation with one molecule instead of 1000 molecules. Then, at given time t, exactly one of numbers ai is non-zero and equal to 1. So the state of our system in the Gillespie algorithm is described by a vector of 40 numbers, which has one coordinate equal to 1 and the remaining 39 coordinates equal to 0. We can extract the position of the simulated molecule from this vector as the index of the non-zero coordinate. I have plotted this number in figure 4.3a to show that plots of illustrative trajectories are also comparable between both models. The results calculated by the compartment-based approach are presented in figure 4.3a, which I have plotted on this slide as the figure on the right. In this case, you can see an obvious difference in the details of the trajectories on the left and right figure, because the positions in the compartment-based model are only known with the compartment size resolution. Since I'm using the centers of compartments in my visualization of each trajectory, these trajectories jump between 40 different values. Other than that, there is not much visible difference between the models of diffusion presented in sections 4.1 and 4.2. And we can prove that. Since the compartment-based approach is formally written as a chemical reaction system, we can use the chemical master equation to analyze it. The chemical master equation describes the time evolution of the probability mass function P, where P and T is the joint probability that there are Ni molecules in the ith compartment at time t. Here, bold N is the k-dimensional vector, where k is the number of compartments. To formulate the chemical master equation, we define two operators. Ri corresponds to the jump to the right from the ith compartment. That is, if the system is in this state and the jump to the right happens, then the system ends up with bold N molecules in compartments. In a similar way, Li corresponds to the jump to the left from the ith compartment. With this notation, it is easier to write down the chemical master equation for our diffusion process. Since our diffusion system is a system of 2k-2 reactions, we will have twice as many terms on the right-hand side of the chemical master equation, corresponding to our chemical reaction system 413. Each reaction contributes to two terms on the right-hand side of the chemical master equation. The first sum corresponds to k-1 jumps to the right, and the second sum corresponds to k-1 jumps to the left. Having formulated the chemical master equation, we can use it to analyze the compartment-based model of diffusion. Since our chemical system 413 only contains first-order chemical reactions, we can use the method from chapter 1 to calculate evolution equation for the average quantities, like the mean and variance. To do that, we define bold M as a k-dimensional vector, where the ith coordinate, mi, gives us the mean number of molecules in the ith compartment. Multiplying the probability mass function P and T by Ni and summing over all possible values of all k coordinates of vector n, we obtain the mean mi. To obtain the evolution equation for mi, we multiply the chemical master equation by Ni and sum over vector n, which we define as summing over all possible values of all coordinates of vector n. I left this calculation as exercise 4.2. It is another exercise to practice the approach presented in chapter 1, which we used for the analysis of first-order chemical reaction systems. The derived equations are equivalent to the classic deterministic ordinary differential equation model. This is not so surprising, as this is a general property of any first-order chemical reaction system. Strictly speaking, the derived system is a system of ordinary differential equations, so I could write here standard derivative rather than partial derivatives. However, I have put here partial derivatives because we will further analyze this system to derive the diffusion equation, which provides the classic deterministic description of diffusion. It is written in terms of concentration Cxt, which can be approximated in the center of the ith compartment by mean mi divided by h. I have written here this approximation symbol rather than an equality because we only have information about the concentration with the spatial resolution of the compartment size h. We can assume that concentration is constant within each compartment, or we could extend these values by some interpolation to define the concentration Cxt for all real values of x. However, other than that, there was so far no approximation in our analysis of the compartment-based model. For example, equations 424 for the mean mi provide an exact description of the time evolution of the averages. Dividing equations 424 by the compartment size h, we obtain the equivalent equations for concentration C at each point xi. By Taylor expanding the right-hand side, we obtain the diffusion equation for concentration C with diffusion constant d, provided that the rate constant lowercase d of our chemical system 413 is chosen to be equal to diffusion constant d divided by the compartment size h to power 2. This is exactly the formula for 13 which we have used in our illustrative simulations. The compartment-based model can be further analyzed. Since this is a good practice of the methods which we presented in chapter 1, I have left some analysis to you in exercises. In exercise 4.3, you are asked to analyze the stationary diffusion master equation corresponding to the chemical reaction system 413, which can be obtained by putting the time derivative equal to 0. You are asked to find the corresponding stationary distribution and stationary values of the mean and variance vector and stationary covariance matrix. To verify your solution, you could check three things. First, you could check that your answers are steady-state solutions of ordinary differential equations for means and variances, given as equations 424 to 425 and equations 430 to 432. You could also implement the Gillespie algorithm for our diffusion chemical system 413 and estimate the averages using long-time stochastic simulations. You could check that you obtain the same mean and variance vectors as you get by the theoretically derived formulas. Finally, you could check that in the case of two compartments, that is for k equal 2, you will get a binomial distribution, which is a standard result for a reversible reaction A goes to B and back. This brings me to the end of lecture 13, where we discuss the compartment-based approach to diffusion. Please read pages 101 to 107 of the lecture notes before watching the video of lecture 14. In our next lecture, we will continue in our discussion of chapter 4, introducing velocity jump processes. Thank you very much for listening to lecture 13 of our course on stochastic modeling of biological processes. Bye bye.